Okay, IB, let's take a look at how the membrane of our cells actually regulate things coming in and out. Sometimes it takes some energy, but sometimes it simply is very passive and can happen somewhat naturally on its own. So, looking at this assessment statement, we want to explain passive transport across membranes by simple diffusion and facilitated diffusion. And so passive transport implies that we don't need energy to move a particle from one side of the membrane to the other. However, it's driven by a concentration gradient. We'll talk about that in a moment. So if you think about if mom and dad or somebody is making breakfast for you on a Saturday morning and you wake up to the smell of breakfast, well, those molecules of the bacon or the eggs or the things that you're smelling, or maybe some really sweet waffles, have basically diffused from the kitchen, where we have a really high concentration of these smells, and they're making their way maybe up the stairs into your nose, or maybe around the corner, wherever your bedroom is. And so they're moving down their concentration gradient. They're moving from an area of a high concentration of a smell to a low concentration of a smell. And so that in itself is diffusion right there. It's just the diffusion of these particles of food. And so diffusion and osmosis are both a passive process. They're very similar, but they're specific to either water or looking at different solutes. Diffusion happens freely across the membrane if the membrane is actually permeable to that substance. And so some things, some gases like oxygen and carbon dioxide, they flow freely through the plasma membrane, but other things like water, they can go through the membrane, but they actually have to have help. They have to go through facilitated diffusion. They have to use a protein to actually pump through. And diffusion in one direction will not stop something from going in the other direction. So if you have two molecules on both sides trying to diffuse through, they will naturally diffuse through, but they're going to go down their concentration gradient. And diffusion allows for the exchanges within the environment and the regulation of water content so that our cells can function, our cells can maintain homeostasis. So simple diffusion is the net movement, which is passive, of molecules along their concentration gradient. And so what that means when we say along the concentration gradient, I am saying it's moving from a high to a low. Moving from a high concentration to a low concentration is easy, and it doesn't require any energy. And so the rate of diffusion is actually going to be higher if the concentration is higher. If you are all clustered inside of a car, right, too many of you inside of a car, and you're trying to get to a restaurant to hang out, you naturally eventually are going to get sick and tired of being so close together that you want to kind of just move out of the car and have your own space. And so that's kind of an example of moving from high to low concentration. And so this doesn't require any help from any proteins or any energy for it to happen, and these molecules will easily diffuse through our plasma membrane. So our oxygen molecules and our carbon dioxide molecules, they are small enough, and they have no problems with the attractions that they have toward this lipid bilayer, that they're actually going to go through and diffuse right through that layer. So facilitated diffusion, it's still passive, but it can't, these are particles that will not go directly through the plasma membrane. So if you look here, here's normal diffusion. These molecules are going through our lipid bilayer, but there are some molecules that are too big, or they have hydrophobic properties. They're not attracted to the phospholipid tails of the plasma membrane, so they're actually going to go through a carrier protein that will help them out. These proteins are going to actually be embedded in the plasma membrane, and remember that these molecules that can't necessarily go through the plasma membrane, it's due to the fact that that membrane is selective. The properties of these molecules actually dictate if they're going to go through the membrane. So like I was saying, for example, water is hydrophobic. It's not going to go through that plasma membrane. It's going to use the help of a protein carrier. A lot of the insides of our protein carriers or protein channels are typically polar, And if another molecule is polar, and so is the inside lining of that channel, that protein channel, sorry, it will actually be attracted to go through. And so we see here, we have two examples of facilitated diffusion. They are both using a protein. And in this case, this one right here is what we call a channel protein. And this one right here is a carrier protein. And so the difference between these two is that our channel protein, it's really just a tube. It's just like a tunnel that allows for things to go through. And our carrier protein is one that is actually going to change conformational shape and carry that protein across the membrane. Osmosis is a very specific type of diffusion, and it's facilitated diffusion. We're looking at water molecules that are actually going to move. It's moving from a low solute concentration to a high solute concentration. So solute is something that is going to be dissolved by a solvent, and in this case water is the solvent. And so this cannot happen through the plasma membrane, because the plasma membrane is hydrophobic because of those lipid tails of that phospholipid bilayer. And so what will happen is water molecules may get stuck, as you can see in this picture, but the reality is they're never going to move. So they have to go through this protein, this protein channel, and it's called an aquaporin. So it's the movement down a concentration gradient, but the movement we are talking about is water, and so that technically depends upon the solute concentration. So even though I'm talking about a low solute concentration, the reality is if there is a little bit of glucose on one side, or sucrose, the reality is there's actually a high concentration of water on that side, and it's vice versa over here. So if the solute is high of glucose, it just means we have a really strong concentration of glucose, therefore we have a low concentration of water. So water moves technically from a high concentration of water to a low concentration of water. This is just a different way to phrase what that statement means. So if you look here, this is a U tube, and the U tube has a semi-permeable membrane right down the middle. It does not allow for these big molecules, maybe these are glucose molecules or a sucrose molecule, it doesn't allow for them to pass through, but water is permeable to this membrane. And so you can see on this side of the tube, the left side of the tube, we have less solute, okay? We have a lower concentration of solute, but what that means is, like I said before, it's a high concentration of H2O. It's opposite on the other side. You have a lot of solute, a high concentration of solute. And what that means is you've got a low concentration of water over here. So water is going to naturally go from a small concentration of solute to a high concentration, which is really the movement of a high to a low.